Hello, good afternoon, everyone. Welcome to our HOPP Summer Research Seminar Series. We are delighted to have you all join us this afternoon. If it's your first time joining our seminar, my name is Vicki Bowden. I am a project manager here in HOPP. And before we begin and I introduce Dr. Dunbar, I'd like to share some information with you all. The first, our Q&A session. If you have any questions, please use our Q&A feature here on Zoom. You'll submit your question and we will address them towards the end of the seminar. Any questions we're unable to answer, Dr. Dunbar will be responding to them via our Google form, and we'll send that information to all registrants after this session. And you'll be receiving an attendee survey following the seminar to provide feedback as well. All of our seminar recordings, we welcome you to visit our YouTube page, HOPP Summer Student Program, and you can view past seminars that you were unable to attend live. So this afternoon, we'd like to welcome Dr. Andrew Dunbar, and he will be speaking on oncogenes and cell signaling. Dr. Dunbar is a laboratory-based physician scientist focusing on the study of myeloproliferative neoplasms and acute myeloid leukemias. His goal is to use animal modeling and state-of-the-art genomic techniques to uncover the genetic and epigenetic underpinnings of these diseases in order to find newer and smarter ways to treat them. His objective is for these findings to be translated into novel therapies that improve the outcomes of people affected by these conditions. Again, thank you.
And thank you, Vicki, for the intro, and for everybody for joining, and for the organizers for giving me the opportunity to speak today. It's a privilege to be able to, talk a little bit about science and cancer research, and maybe a little bit about myself. And the goal today is sort of to give you a very brief primer on cell signaling and oncogenes and signal transduction pathways and when things go awry in cancer. I figured I'd just talk a little bit about myself up front here. So as Vicki said, I'm a cancer doctor. I have an MD. I have a particular interest in leukemia and other blood disorders and the treatments of these diseases. I work in a lab mostly. I work in Ross Levine's lab here at HOPP, studying leukemias and blood disorders, mostly using mouse models of these diseases to uncover biology and hopefully to develop new treatments. But I also have a clinical responsibility. I take care of patients on the inpatient leukemia service in the hospital when patients are admitted for either a new diagnosis or a treatment or complications of that treatment. And again, as Vicki said, I'm sort of interested in how gene mutations contribute to leukemia growth, with a particular focus in epigenetics, sort of the winding and unwinding of DNA to control gene expression, and how these mutations can sort of affect response to therapy, and perhaps more importantly, how we can overcome those things with newer and better drugs. And I'll just briefly mention something about mentorship, which I think others had commented on in the past, because I think it's an incredibly important thing. You're at such an important stage of your lives. Sort of beginning thinking about what you may want to do for your careers long term. And I have been so blessed and lucky to have incredible mentors at each and every step along the way, from early sort of getting in the lab, working with a pipette in college with Dr. Singleton, to working as a research technician at Cleveland Clinic under Jarek Maciejewski, who really allowed me to take on individual projects. And that's really sort of when my fascination in cancer research and cancer biology really bloomed. And then through med school with Dr. Carmen, and of course now here in Dr. Levine's lab at Sloan, this really is sort of the MBA, the pinnacle of cancer research. And to have the opportunity to work with Dr. Levine and to see sort of how he sort of shapes and molds junior faculty and recruits and people into his lab it's very inspiring, and like I said, I'm very lucky to be able to have the opportunity to work in this environment. And I think, again, it's just if you can find those people in your life who are going to invest in your development and motivate you positively in the things that you're very passionate about, it almost becomes sort of a self-fulfilling feed forward effect where everybody's sort of benefiting everybody. So again, it's the importance of finding those people who are going to help get you to the next step. So today, the title of the talk was Oncogenes and Cell Signaling, which is a huge topic, decades worth of research with, an innumerable number of influential names and leaders, of which people have won Nobel Prizes in this sort of thing, and is a semester's class, if not a years-long class worth of material to ultimately get through. Please understand that this is very much scratching the surface, but I hope at the end you'll have a basic understanding of what cell signaling actually means and when this goes wrong in the development of cancer. And so the sort of outline for the talk today is, first of all, simply to define what that means, cell signaling, also known as signal transduction, and talk about some of the sort of canonical critical signaling pathways in the eukaryotic cell, with a particular focus on the Ras-Raf pathway. And then when those pathways go wrong, and when they are hyperactivated in cancer, which and contributes to cancer growth, how those pathways then can be potentially targeted with anti-cancer drugs, and then the potential consequences of that therapy. And then if there's time at the end, I can maybe show you a little bit about the stuff that I'm working on in the lab, which I think sort of goes hand in hand with cell signaling. So it's been a long time since high school. I'm old at this point. I'm not afraid to admit that, or getting old. And so I was thinking about sort of back when biology and sort of what we were learning at the time to sort of frame the whole the context of everything. And I thought back very much to my 8th grade biology class, when you first learn about the eukaryotic cell and its various sort of intracellular organelles and the structure and function of these organelles. And I remember being that age sort of realizing for the first time just how complex these sort of seemingly simple single cell organisms really were. And, when you go to college and you take more advanced courses in biology, you sort of begin to further understand the vast complexity of the cell itself and the environment within which it lives. And you come to appreciate sort of what they call as homeostasis of the cell, this sort of very highly dynamic, complex equilibrium that the cell exists in, where it's sort of constantly receiving signals either from its immediate vicinity or from a distant location in a multicellular organism, or from within, from its own cell, and taking these signals and initiating intracellular processes in order to receive and understand the signals. And by that I mean complex interactions in regard to its cell survival and growth and metabolism and its movement. And I just have this just to show that the cell exists in normal homeostasis, but is frequently encountered with various extracellular stresses to the point where sometimes it's almost injurious. And because it's such a highly dynamic state, the cell is very good at sort of compensating and dealing with these stresses, either to the point where it can sort of reverse the injury and go back to a normal hemostasis, or if it's so bad, undergoing a state where it essentially is going to kill itself off. And again, I just put this up here just to show you that the cell is constantly existing in this highly dynamic equilibrium state in order to survive. And at the heart of this, at the crux of maintaining this homeostasis, I feel, is cell signaling and signal transduction. And I think this is sort of a very simple schematic that gets to the heart of actually what this means, in that here you have sort of this diagram of the cell. You have this sort of extracellular space. You have the cell membrane here with this, eye to represent a protein that sort of lives in the cell membrane, the cytoplasm here, and then here's the nucleus again, surrounded by its own sort of nuclear membrane and DNA sort of existing in the nucleus. And cell signaling is the process by which you have these sort of extracellular small molecules called ligands that are in the vicinity of the cell and are recognized by these proteins that exist at the cell membrane. They're called receptors. And when the ligand binds to these receptors, there's a conformational change in the shape, the actual physical shape of the protein that leads it into an active state. And that in turn activates other proteins existing on the inside of the surface of the cell membrane, which in turn causing them to become active, which in turn causes additional downstream proteins here to become active, which in turn again causes further downstream proteins to become active, ultimately leading to entry of these proteins into the nucleus of the cell, where they bind to transcription factors and regulate gene transcription and the production of RNA that is ultimately translated into proteins that are critical for cellular processes. I should also mention that this is not the only method ligands can sort of communicate signals from outside to in. You can have ligands that sort of pass through the membrane and bind to intracellular receptors, but at the end of the day, the key point is that you're taking a signal from outside the cell, transmitting it through the cell through a series of various proteins to ultimately influence change at the genetic level by gene transcription and protein production. And again, at the heart of this whole process are the essential processes that keep the cell alive in terms of cell proliferation, so the growth from one cell into 2, into 4, into 16, so forth. Its survival, its metabolism, its ability to make energy, its ability to sort of move and interact with cells nearby. And so I can't stress enough of how important cell signaling is at communicating and amplifying the signals from the area nearby. I should also say, though, that this is a profoundly simple view of signal transduction. Really, at the end of the day, there isn't just one pathway, there are many, and there isn't just one receptor on the surface, there are many, and you have a lot of pathways and a lot of crosstalk between pathways. And I sort of suggest here that there's sort of an amplification activation of effect. However, there are also many proteins involved in the negative regulation of pathway signaling that are designed to turn off the pathway. But the takeaway from all of this, again, is to say that it's a highly complex network that keeps the cell in an equilibrium state in order to influence its survival. Perhaps the most well-described pathway known, and particularly in cancer, is the Ras-Raf mitogen activated protein kinase pathway. It's sort of consists, and pathways are sort of named by the major proteins that operate within it. Here's sort of a simple schematic of the Ras-Raf pathway. And again, the principle is the same. A ligand binds to a receptor on the plasma membrane. There's a series of protein cofactors that Ras sort of comes and binds to and becomes activated. That in turn activates downstream RAF, which in turn activates downstream MEK, ERK, and then ultimately gene transcription and the cellular functions that I've already talked about. The energy that RAS and RAF and these other proteins use to activate downstream effectors is this GTP molecule, this guanosine triphosphate molecule. When GTP binds to KRAS, it becomes it comes to an active state with active signaling. And when a phosphate group is removed and it becomes guanosine diphosphate or GDP, KRAS is inactivated, and the pathway is shut off. The point of this is just to say that there's a switch to these things, that the pathway is at some times on, and then there's a pathway, and then there are times when the pathway is off. And this is essential, again, for normal cell functioning. Which is now why you can imagine where things can go wrong in these pathways. And so, and this brings us to the definition of what an oncogene is. An oncogene is a gene that encodes a protein that, when mutated, promotes the development of cancer. And so in that regard, you can imagine then that any one of these proteins in the pathway, from the receptor on the surface of the plasma membrane to the transcription factor that binds DNA and affects gene expression, is a potential oncogene. And that's exactly the case. So, for example, RAS itself is frequently mutated in cancer. And when that happens, you have constitutive hyperactivation of all of its downstream targets. And this leads to aberrant cell proliferation, growth, and survival, and all of these things that, sort of had already mentioned about. And this is sort of makes the the classic way in which cancer is thought to develop, in that you have a single mutation of one of these oncogenes in a single mutated cell that leads to aberrant cell growth that over time continues to grow and become more aggressive, ultimately leading to an invasive cancer that unfortunately at sometimes can spread to distant sites and become metastatic. And so RAS, as I said, is very, very frequently mutated in a multitude of different tumor types. I should say that the human body has 3 different types of RAS genes: KRAS, NRAS, and HRAS. KRAS specifically is probably one of the more frequent mutated RAS genes in cancer. And this is just a bar graph showing the frequency of KRAS mutations in the different sort of tumor types shown here along the bottom. And you can get a sense, for pancreatic cancer, for example, over 50-60% of patients have mutations of KRAS. That's a striking amount of patients who have mutations of this gene. And you see sort of similar frequencies in pancreatic cancer, frequent KRAS mutations in non-small cell lung cancer. So this is a critical gene and clearly a critical pathway, again, involved in the cellular growth and survival, of these basic functions. And so what does it mean then for an oncogene to be quote unquote mutated? This, I think, is very interesting for KRAS specifically and other oncogenes, in that you actually have a single change in one nucleotide of the DNA of the DNA of the KRAS gene that causes this aberrant cell growth. And this is sort of what I'm trying to show here on the left. So each of these different peaks here, this is Sanger sequencing, and each of these peaks represents a single nucleotide within the KRAS gene around the site of the mutation. And these are sort of your individual nucleotide bases that you might have learned about, A, G, T, C, and so forth. But you can see here at the arrow, what once was a single black peak, you now have this green peak and this sort of nucleotide change of a guanine to an adenine. And this results in an amino acid change in the KRAS protein from a glycine to an aspartic acid. And when this happens, there's a structural change in the KRAS protein itself, shown here sort of in the inset, that whereas with wild type, you have this ability to lose GTP to GDP and turn RAS off. In the setting of the mutation, it doesn't turn off. GTP cannot be removed, and there's persistent activation of the pathway downstream of KRAS, represented here. And then again, you have aberrant overactive signaling of this pathway, aberrant gene expression of the Ras-Raf gene targets, and then that results in tumor growth. And I'm just showing this this is what they call a Western blot. This is sort of one of several of many tools that you can do in the lab to look at the extent of aberrant pathway activation. And you could see here for like p-ERK and p-MEK, for example, that the normal is less here compared to the mutant RAS shown here or here. This is just to show that there is a more abundant amount of active MEK or ERK in mutant RAS cells than there are in normal cells, just confirming that truly these pathways are hyperactivated in mutant RAS cells. I was just going to mention something also about nomenclature. So you might hear things, these point mutations are called, you might hear them described as G12D, for example, a letter, a number, and a letter. This is a frequent nomenclature used by cancer biologists to describe point mutations. Basically, it means that, here's your gene that you're talking about. The first letter is representative of the wild type amino acid in the protein, so glycine. 12, the number, is represented by, it represents the number in the amino acid chain of the protein that's changed in the setting of the mutation. In this case, it's the 12th amino acid of the gene, of the protein out of 189. And then the last letter, the D, represents the mutant amino acid change. So the glycine has switched to an aspartic acid at the 12th amino acid. So this is the KRAS G12D mutation. And again, this is sort of standard nomenclature for point mutations in cancer. KRAS, as I said, is just one of many different oncogenes mutated by point mutation in cancer. BRAF V600E being another one, JAK2 V617F, PI3 kinase, and so forth, but use the same sort of standard nomenclature in case you see that sort of going forward. But point mutations, so these single nucleotide changes, are just one of several ways that oncogenes can become activated and mutated in cancer. Another common example is what they call gene amplification. So the proto-oncogene, sort of the wild type form of the gene, so KRAS, for example, is amplified and duplicated. And so you, instead of having just one KRAS, normal KRAS gene, now you have 3. And simply by a matter of having more abundant wild type KRAS, you have an aberrant increase in RAS signaling. RAS is amplified in many cancers. Other examples of genes amplified in cancers are MYC and HER2, for example, in breast cancer. In addition to gene amplification, you can also have what they call translocations. So big chunks of chromosomes are essentially broken off from where they normally are and replaced or moved to other chromosomes in the cell, and resulting in regulatory regions of one gene affecting the expression of your oncogene. And so, for example, a prime example of this is the BCR-ABL fusion, where ABL1 is sort of moved into close proximity of the BCR gene, which is overexpressed and overexpresses the ABL1 gene as a result. And so you have, again, the ABL1 gene has the normal DNA sequence, but simply because it's overexpressed, you have overactivation of the ABL1 kinase pathway. I should say then, you could also imagine that, as I said before, that any one of these potential proteins along the pathway is a potential oncogene, in that if mutated, it can activate any and all downstream targets. And again, that's exactly what we see. As I mentioned about before, we see mutations of BRAF frequently in various cancer types. Again, this results in activation of its downstream targets, leading to cell proliferation, growth, and survival. We see BRAF mutations frequently in melanoma. We also see translocations of MYC frequently in lymphoma, specifically, where you have rearrangement of the MYC gene up against the immunoglobulin locus, leading to MYC overexpression, which in turn, again, leads to overactivation of downstream targets, cell proliferation, growth, survival in lymphoma, specifically Burkitt's lymphoma. We see activating mutations in PI3 kinase, very common in breast cancer and in uterine tumors. Again, overactivation of the pathway downstream of PI3 kinase, leading to aberrant cell growth. We see frequent mutations in the receptors on the cell surface as well. These are very common in cancer, e.g. the epidermal growth factor receptor, the HER2 receptor, the ALK receptor, c-KIT, again, frequently mutated in cancers that lead to constitutive signaling downstream of the receptors, again leading to cell proliferation, growth, and survival and cancer. So I should say that, interestingly, the majority of these mutations, particularly point mutations, really weren't well known before the advent of more sophisticated DNA sequencing techniques. So it really was in the early 2000s, probably around the time where many of you guys were born, where capillary sequencing was really coming about. And for the first time, the ability to sequence vast amounts of DNA was becoming increasingly plausible and possible, and also inexpensive. And so this allowed us to really sort of explore the genomes of the tumor types to identify the mutations that might be contributing to disease development. And so with this, there was an explosion, really, of very high impact papers exploring, again, the genomes of these tumors and various tumor types in breast cancer, lung cancer, colon cancer, liquid tumors like leukemias and so forth, PI3 kinase and BRAF and JAK2, sort of all discovered around this time in the early 2000s, again, with the advent of more sophisticated DNA sequencing technology, and sort of really blew the roof off sort of our understanding of what can cause cancer and what can go wrong, and really sort of got people starting to think more about how we could better target these cells to treat the tumor. And so you could sort of imagine a miracle drug, a mystery drug that was capable of binding to the mutated protein, in this case, for example, the receptor, and effectively shut it down. And in doing so, you shut down the pathway, and in principle also shut down tumor growth. And this was 20, 25 years ago now, and nowadays targeted therapy is sort of ubiquitous and is part of standard guidelines for the treatment of multiple different tumor types. But at the time, this idea was met with a lot of skepticism, because there was a thought of alternative signaling or other targets contributing to cancer growth beyond these individual proteins, let alone compounds that would actually be able to bind with high affinity and efficacy to these mutated proteins. So it was really met with a lot of skepticism, as I said. However, this really sort of all changed when work by, again, Charles Sawyers, who sort of runs HOPP here, along with Brian Drucker and pharmacologists elsewhere, started to explore this idea of targeted therapy in a liquid tumor called chronic myelogenous leukemia. This was prior to, in the 1990s and prior to the 2000s, could be a very devastating disease for which there weren't a lot of effective treatments. This is just a representative smear of a patient with CML, and you can get a sense of just how high these white blood cell counts are in these patients, and sort of the effect that high white counts like this can have in terms of their symptomatology and so forth. And CML sort of follows generally a chronic course, which sort of gives its name. But eventually, over time, it unfortunately, inevitably leads to what they call blast crisis, which the cells become very immature and aggressive and fast growing, and mortality in these instances is very high. And like I said, prior to the early 2000s, the treatment for these were standard cytotoxic chemotherapy, the chemotherapy that's sort of been around for decades, in addition to hydroxyurea and interferon, sort of other treatments that were really sort of a drop in the bucket in terms of what was required to actually treat this disease. But it was also around this time that it was becoming increasingly clear about the biology of what actually caused CML, and it was found by several groups that it's really this BCR-ABL1 fusion protein that I sort of mentioned about before that is driving the disease persistence. So the BCR-ABL gene fusion protein is found in 100%, I would say, of patients with CML. And there have been, again, excellent work in the past showing that BCR-ABL1 and BCR-ABL1 alone is sufficient to induce a chronic myelogenous leukemia-like disease in either cells or in mouse models. And so this really is the causative factor for this particular disease. And as we've sort of talked about in the context of cell signaling, this leads to aberrant ABL1 kinase activity and an overactive downstream signaling of many of the pathways that I've sort of already mentioned about, including PI3 kinase, AKT, the RAF MEK MAP kinase pathway, as well as JAK-STAT, which we didn't really touch on. But with persistent activation leads to the proliferation and growth of these leukemia cells. But the thought is that, again, if there's enough data to suggest that it's ABL1 kinase alone that's driving this disease, can we develop a compound that could potentially inhibit it and therefore shut these cells off? And through various drug screens and work by Brian Drucker, they identified a potential promising compound that showed activity in preclinical models, enough to push this compound into a phase 1 clinical trial in the late 1990s. And the name of this compound was imatinib, also known as Gleevec. And how it works is that it actually binds it's a very small molecule and binds to this ATP binding pocket in the BCR-ABL fusion protein. And basically, it prevents ATP from using ATP to activate downstream effectors. And so you essentially if this was RAS, for example, this complex right here, it's essentially not turning it off into an inactive form, so that all of the downstream effectors, if this was RAF, for example, is shut off and the pathway is shut off entirely downstream. And so it was sort of a novel idea, these little small molecules that bind with high specificity to certain areas of these compounds. And what was so striking about this trial with this drug for patients with disease was the absolute incredible, almost unheard of response rate of 100% at sort of the dosing level that we currently dose this drug at. 54 out of 54 patients responded. And these were patients with CML in chronic phase, and just to see the counts normalize must have been very striking to see in all of these patients. And interestingly, they could actually also check the amount of the BCR-ABL fusion transcript in the peripheral blood of these of these patients. And they show that, over time, within 100 to 150 days of being on this drug, the presence of these mutant cells, these CML cells in the blood of these patients dropped to levels near zero, which was striking and a testament to how effective this drug really was for this particular disease. And obviously, this had big implications for clinical outcomes with patients with CML. This is sort of a classic Kaplan-Meier curve of survival for patients based on the respective treatments that they were receiving. And you can see out to 72 months for patients who were receiving this Gleevec drug, over 90% of the patients were still doing well in comparison to, unfortunately, the patients who had received sort of more standard treatment. And so Gleevec and imatinib and subsequent, second generation and third generation ABL1 kinase inhibitors have really changed the landscape of CML treatment. And it's sort of a testament to Dr. Sawyers and Dr. Drucker and Dr. Leiden for pushing the field forward and leading to these sort of remarkable results. And I'll just say that, as a physician, as a leukemia doctor who actually takes care of CML patients, it's actually quite striking to see these very abrupt and immediate responses, and to trend the BCR-ABL fusion transcript in the blood of these patients over time. And I just show this as a curve because it's part of the actual standard treatment guidelines to monitor this transcript in the blood of these patients and expect that the amount of disease cells to drop to near undetectable levels. And again, it just sort of goes to the potency of these compounds in treating this disease. And so, again, this led to a boon and a big development of additional targeted inhibitor therapy. This is a large list of a lot of the additional targeted drugs that are either currently being offered in the clinic or are still under development. But you can see sort of here the different targets that they go after, components of the Ras-Raf pathway, as I mentioned about. You have MEK inhibitors, you have RAF inhibitors, inhibitors of the kinase of the receptors on the surface, EGFR, VEGF, and so forth, the BCR-ABL inhibitors. So there's a whole host of targeted therapies now directed against these mutant oncogenes in order to shut down their activity. And it's, again, changed the way we sort of thought about treating patients. And we're very much in this era now of precision and precision medicine and personalized medicine. And this is very much to the heart of what we do here at Memorial Sloan Kettering, where new patients coming to the hospital for an evaluation we take their tumor and we essentially sequence it right away. And we can get a panel of mutations found in the tumor from each individual patient. And then we oftentimes can select the treatment that they get based solely off the mutations that we find in their tumor, with the thought being that we can sort of, with a smart bomb, target their tumor cells directly and then monitor over time, the response to these targeted treatments. I should say, though, that it's not a perfect system. Cancer is very good at finding ways to evade these therapies, and we now have a very good understanding of how tumors can develop what they call resistance to target inhibitor therapy. And this is sort of just a cartoon demonstrating sort of what I mean by that, in that if this is your tumor here, sort of before any sort of treatment, you might have the vast majority of cells that respond very well to a targeted inhibitor that you give. However, it's possible that you might have even one cell out of a million that, for whatever reason, has a change in the DNA or something in the metabolism that renders it unresponsive to that particular targeted treatment. And so that when you treat a patient with this drug, yes, you might have eradication of 99.9% of the tumor cells, but you might have one or two that persist because of these changes, and that over time, even with continuing to take the drug, you will have an emergence of what they call this sort of resistant clone that, despite continuing to treat with the same drug, will never respond to it. And so a relapse occurs, and you have to think about alternative therapies. And so this is exactly what happened in the case of BCR-ABL. In the case of BCR-ABL and imatinib, you had a single, like the KRAS point mutation, activating point mutation, you had a single point mutation in BCR-ABL that actually prevented the drug from binding to its normal target. And when that occurred, BCR-ABL then was allowed to become active, and you would have relapse or reemergence of disease. And so you have to think about alternative, inhibitors or other agents to treat the drug or treat the disease. And I should say that this isn't the only mutation in BCR-ABL that can lead to resistance to imatinib. There are a multitude of other different point mutations occurring in the BCR-ABL fusion transcript that can lead to lack of responsiveness to imatinib therapy. And so we're sort of always trying to play catch up with newer drugs that bind with higher degree or elsewhere, next generation inhibitors that will overcome imatinib resistance and so forth. And this isn't true just for CML. We see this in multiple tumor types with multiple target inhibitors, EGFR inhibitor resistance in lung cancer, so forth. And so, what are the potential things that we could consider to overcome this, or what is sort of the newer drug compound on the horizon that might better target these oncogenes to shut off these pathways? And so something of interest to me are these, what they call these proteolysis targeting chimeras. These are compounds still very much in infancy, but they're sort of up and coming. And the basic gist of it is that here in green you have your mutant oncogene, let's say Ras, and existing in the cell. And then you have this E3 ligase protein. This is part of the normal cell operation. It's a complex of proteins designed to degrade proteins that are of no longer use to the cell. And so the thought is that if you can design a compound, these PROTACs, that link your mutant protein to this sort of degrader complex, you can bring them together and tag the mutant protein for degradation. And the degradation sort of is done in a very controlled manner in these sort of organelles called proteasomes. And this leads to, basically the protein getting, broken up and released as simple fragments. And so you could imagine a scenario where if you had a drug that was specific enough to mutant Ras, it would bind with high specificity, get rid of it entirely, and shut the pathway downstream off, and hopefully control tumor growth. And so this is, again, an idea that's still very much in infancy, but something sort of coming up on the horizon that is sort of I'm particularly excited about. This is actually a paper that was published to bioRxiv, so it's hot off the presses. This is a preprint, not even officially published yet, showing an example of a KRAS degrader. The key point that I want to show is just this Western blot here, where if you look at these bands, these dark bands, as they're adding drug here, shown in red with these numbers, and at increasing doses, this band gets lighter, representing that the KRAS protein is actually going away. It's effectively being degraded, presumably. And that, interestingly, this coincides with loss of phospho-ERK, so the active form of ERK downstream of KRAS. So not only are we seeing proof that KRAS is being degraded at increasing doses of drug, we see that the pathway downstream is being shut off. And so obviously there's a lot of caveats to these compounds. There's a lot of still work to be done, but it's something of interest, and who knows, within the next 5-10 years, we might see an increasing number of these in trials in our clinic. I have 2 minutes, and so if you don't mind, I'll briefly talk about some of the work I'm working on in the lab, because I think it's a little relevant to oncogenes and oncogenic signaling. So I study this disease called myeloproliferative neoplasms. It's, again, in sort of another blood disorder where you basically get abnormal proliferation of mature blood cells. So you get high platelets, high red blood cells, and then sometimes you get high mature white blood cells that can cause scarring in the bone marrow. And I think it's relevant to today because the vast majority of patients with this disease, MPN, have mutations of JAK2 V617F. And this is sort of the pathway here. You have mutations here in JAK2, and it leads to, as we've already sort of talked about and emphasized, overactivation of downstream targets and aberrant cell proliferation, growth, and survival, and the development of disease. And we've developed inhibitors of JAK2 that bind JAK2 to shut it off and shut down the pathway. Ruxolitinib has been in the clinic for years and is very efficacious for the treatment of this disease. However, in contrast to ABL1 kinase mutations in CML, patients who are receiving ruxolitinib for this disease, their mutant cells don't go away. They persist in the blood despite being on the drug. And there's a big question in the field of why that might be occurring. And the Levine lab has looked at this in the past, and there's convincing evidence to suggest that you actually have activation of pathways around JAK2 as a result. And so the question is can we design better inhibitors of mutant JAK2 that can eradicate the mutant cells just as it was with imatinib and CML? And so that compound doesn't exist. And so what we've been doing is using mouse models to do the next best thing. So this is very confusing, and I don't need to go into it in depth, but I want the takeaway from this slide to be that we've designed a mouse, basically, where we can turn on the mutant JAK2 gene and get a disease, MPN, and then at a later time point, delete the gene from the mouse entirely. And so effectively removing the gene from the genome of the mutant cells and seeing what happens. And in many ways, replicating or modeling the ultimate inhibitor, right? Because if you can actually delete the mutant gene from the genome of the mouse, you can't get a better inhibitor than that because it's not there anymore. And so what we've found, interestingly, is that when you do delete JAK2 after knocking it in, lo and behold, and perhaps reassuringly, the blood counts normalize, shown here on the left. But not only that, the mutant cell fraction in the blood of these mice goes way down to near zero by 24 weeks. And that's true whether you delete the gene sort of early on after disease starts or later after disease has been around for a while. And so we think that, maybe this data would suggest that theoretically you could design a better inhibitor than the currently available ruxolitinib to better target these cells. And so that's pretty much it. That's a lot in a short amount of time. I hope some of that made sense. I'll just recap by saying that these signaling pathways are absolutely essential for the maintenance of the life of the cell. And they are designed to transmit signals from the cell surface and from other parts of the cell to the nucleus, where they influence gene transcription and critical cell programs in order to maintain their survival. And that many signaling pathways exist. Ras-Raf, I think, being one of the more well-described ones of them. But at the end of the day, they really converge on many of these critical cellular programs. And that oncogenes are, genes that, when mutated, cause overactivation of these pathways, contributing to cell proliferation and cancer growth. But that we now have many inhibitors of these particular proteins in our armamentarium to treat many of these cancers, but that we're unfortunately encountering resistance often with these drugs, and resistance is common, and so we need newer and better and smarter ways designed by people who are up and coming, such as yourself, to help us further better treat patients with these terrible illnesses. So with that's me. Hopefully this was interesting, but happy to take questions, and you have my contact information there as well if you wanted to reach out for other things offline, and I'll turn it back to Vicky.
Great, thank you so much, Dr. Dunbar. We do have a couple of questions, and I'll start off with Neha Maskey, and they ask, why does cancer risk increase as we get older?
Oh, that's a fantastic question. There are many things that might contribute to that, but know that mutations in the DNA are occurring all the time as we age, even in children. And there are tens of thousands of genes in the genome, but the vast majority of our DNA is in non-coding regions where it is of no significance. And But as we age, mutations will accumulate with time. And so, it really is a matter, in many ways, as a matter of just simple bad luck and chance of a mutation occurring in a gene in a cell that gives it that particular growth advantage that leads to cancer. But it's as we age, we develop mutations randomly, and that's the reason why we see more cancer in elderly individuals. Part of the reason why.
Great. Salwa Amayuf asked, what is the difference between gene mutations and gene variants and their implication in cancer development or response to anti-cancer drugs?
Gene variants?
Yeah, the difference between mutations and gene variants.
Forgive me, I'm not entirely sure how to best answer that. Because I'm not entirely sure what is meant by gene variants. Forgive me.
Okay, not a problem. And we'll leave that question in the
Yeah, let me think about it. It's a great question. I don't know how to best answer it.
Yeah, not a problem. We'll follow up with that later on. And Rayan Tuzier asks, can a cell that is in homeostasis and normal be ordered to kill itself, such as apoptosis?
Absolutely. So again, the cell is constantly in a state of recognizing signals from its environment. And many things can induce the cell to say, to die, to undergo sort of programmed cell death, apoptosis, often in response to injury. And so it's actually it's built into the cell, this sort of fail-safe mechanism where if an aggressive amount of DNA damage occurs, or the cell, something happens metabolically where the cell cannot sustain itself, it initiates these programs, evolutionarily sort of withheld within the cell, to basically undergo apoptosis and kill itself because the risk that it poses to other surrounding cells or other things is too great, and it can't survive.
Okay, great. We have another question in reference to Gleevec. So why does Gleevec get rid of leukemia cells, whereas JAK inhibitors fail to do so in MPN?
That's a phenomenal, excellent question, and I wish I had a better answer to that. And I'm hopeful that with continued work on my project, I'll be able to better answer that in the future. But it's true. There appears to be certain tumors appear to be more reliant on individual gene mutations than others. CML specifically, I'd say probably more than any other cancer types that I can think of, with the exception of a few, is exquisitely sensitive to ABL1 kinase inhibition, and I think it goes to show you just how reliant then CML cells are on BCR-ABL1 specifically. However, there are co-occurring mutations. There are other lesions that can occur in addition to EGFR mutations or KRAS or whatever that further promote tumor growth, and that in turn can sort of influence response to targeted therapy.
And Aviva Gelano asks, can there be multiple pathways for the same receptor? And if there are, what factors play a role in which pathway the receptor follows? Are epigenetic factors involved?
I mean, again, that's an excellent question, and I think there are so many factors that will influence how and to what degree a particular pathway is stimulated by a specific ligand, by a specific receptor. Like I said, there's a lot of crosstalk between pathways, between cellular systems. So the cell is constantly making energy, and is undergoing metabolism, and so forth. And so those things factor into which pathway is activated over another. an innumerable number of ways that sort of influence those signaling pathways, and to what degree, yeah.
Okay, great. Amalia Zedji asks, what do you mean when you call for a drug to be designed, quote unquote? What chemical properties of, for example, Gleevec, make it specific to target that mutation?
Yeah, again, excellent questions. So there are many ways compounds find their way from the very early stages all the way to the clinic. In many ways, oftentimes compounds are found by doing sort of drug screens. So you have sort of cells in a dish that have the BCR-ABL mutation and grow aberrantly, and then on a large plate, you actually have like hundreds of compounds, and you really don't know which one is going to really affect the cell growth until after you sort of added these compounds to individual wells and then did cell counts afterwards. And that's often sort of how drug discovery can happen, is that you find the one compound that killed the most cells in a dish, and then you sort of reverse engineer. You can go back and sort of look where that particular compound is binding to ABL1 or to your sort of protein of interest. And sort of that leads you down a route in terms of mechanism of action. And then on the flip side, you're also sort of pushing that compound further forward by putting it into model systems such as mice and other animals that might give you some information about the potential clinical utility of the drugs and also toxicity and side effects. Because as much as we want these drugs to inhibit their target and control cancer growth, we also want them to be tolerable to patients. So it's a whole. I mean, it's a multi-billion-dollar industry of drug development. Yeah.
Okay, great. So we have some questions specifically about not necessarily your work and research, but just in general, your career path. So what made you research blood disorders rather than other types of cancer disorders?
Yeah, I appreciate the question. Like many others, I think we may have personal connections to cancer. My father had a blood cancer, and he later passed from it. But it was during that time where he was being treated where I was actually actively involved in doing cancer research. This was when I was at the Cleveland Clinic. And I think my personality style, I'm a very analytical person. I'm a very neurotic person. Everything sort of has to be in a very specific place. And I think that sort of benefits working in a lab where, there's a lot of technical things you have to keep track of at once. And, there's a certain element of perfection of trying to get things to work in a lab. So I think inherently my personality was drawn to doing translational research in a lab. And then, for personal reasons, I sort of found myself in a liquid tumor lab. Yeah.
Okay, great. And the last question is, based on what you can share, what are the next steps in your research? How do you go about designing a better JAK2 inhibitor that could be used in humans?
Yeah, it's a great question. So, and literally, I'm going to go back to the lab and work on this even more. So, it's designing the right experiments that will help get us to those answers. So I have several different transplants lined up with these mice where we can delete away JAK2. I want to sort of compare deletion of JAK2 with ruxolitinib and other JAK inhibitors to confirm that what I'm seeing is true and that it truly does contrast to what we see with ruxolitinib and other JAK inhibitors. I want to look at how deletion of JAK2 affects gene expression patterns, downstream in comparison to JAK inhibitor therapy. Is there a certain pathway that's activated as a result of deletion in comparison to inhibition that could theoretically also be targeted? And then, another large aspect of my research is also looking at co-occurring mutations with JAK2. So I sort of mentioned about this interest in epigenetics. Mutations in the epigenome occur frequently in liquid tumors and co-occur frequently with JAK2 in MPNs. And so the model system that we're using, this mouse system, is designed as such so that we can actually look at how the disease goes away when you have a co-occurring mutation. And there's some interesting data to suggest that in the background of a co-occurring mutation, the disease doesn't go away as well. Mm-hmm. And so it might be irrespective of any sort of inhibitor, it might be other genetic changes that might be influencing the degree to which MPNs rely solely on JAK2 for disease maintenance. So I think the implications for that are much larger. I mean, I hope so, for studying other oncogenes in cancer.
Okay, great. Thank you so much, Dr. Dunbar, and thank you all for submitting your questions. And for those questions that weren't answered, we will be sending the Google form for you to post them there. And
Dr. Yeah, happy to, yeah, of course, happy to do whatever. Yeah.
So before we end, would like to share a reminder with you all. So our next session will be on the 30th, and we'll be having Dr. Guillermo Aguiles Martinez speak on immunology and cancer. So we look forward to you joining us all. And again, thank you for joining us this afternoon, and enjoy the rest of your day.